Okay, so I asked you to list as many prokaryotic structures that, Mr. Nye, that you can think of. And let's see what we all came up with. Paramecium. No, paramecium are a type of protist, so are amoebas. But bacteria and archaea, those are types or domains. No, they don't have feet. Bacteria, okay, DNA loop, there we go. Membrane, flagella, pili, ribosome, cytoplasm, RNA sometimes. Flagellum, membrane, DNA loop, pili, there we go. Don't have a nuclear envelope, cell wall, cytoplasm, RNA, DNA. There we go, see if there's anything new. Flagellum, yep, are you flagellum? Some are multiflagellated. Algae are like multiflagellates a lot of times. Kind of a fun Are you flagellum? Yes, I'm multiflagellated. So today we're going to be talking about prokaryotes. Now I didn't mean to mention algae when I was talking about prokaryotes. Let me remove that from your brain right now because algae are definitely not prokaryotes, but I was talking about flagella. So these include bacteria and archaea, those two major domains and kingdoms, right? Don't forget, those are domains and kingdoms, which are fully functional cells. They are microscopic, which means you need the device in the middle of your table to see them, and they range in size from 1 to 10 micrometers in length. To give you an idea, our cells are around 30 micrometers. Something needs to be smaller than our cell to infect it, and we know we can get bacterial infections. This is why they are smaller. If you don't have your notes out right now, you probably are not doing what you're supposed to be doing. Ryan squared. Gentlemen, get your notes out. I'm waiting. And about 0.7 to 1.5 micrometers in width. That means that they are generally elongated in shape. Okay, they're kind of like I think of them as a pill, like a little gel cap is what they look like in a way. Okay? They're abundant in the air, water, and soil, and on most objects. They're sitting on the table in front of you right now. In fact, when you walk through your yard, there is bacteria there that would kill you if it were in your body reproducing. Okay? And that's okay. It's not in your body, right? It's in your yard. Now, there have been cases where during football games, guys get tackled and they get these nasty infections, and they've killed people from that. I'm not even kidding you, just from bacteria in the dirt. It's normal. It's all around us. We have this protective barrier on us, though, called an integumentary system. That's your skin, right? It's epithelial tissue, and it waterproofs. It coats your body. It keeps the stuff out. You have a stratified layer of epithelial cells that keeps all of that nasty stuff out. So Louis Pasteur, this is when we talk about vaccines, when we talk about fighting infections, this is kind of the father of immunology. He showed that a previously sterilized broth cannot become cloudy with growth unless it is exposed directly to air. So that means that if things are sterile, like a can, right, you kill everything and then you seal it, nothing can get in, nothing can get out, nothing's going to grow. Okay? Kind of a no-brainer to us today, but back then it was revolutionary. Okay? This helped people develop new sterile methods of keeping things fresh, of preventing infection, increased shelf life of our food, you name it. So prokaryotic structures lack a membrane-bound nucleus. You guys told me that. It's nothing new to us. That means DNA in a nucleoid region. So we call it a nucleoid region because it's the area that would contain a nucleus but does not. It contains just the DNA. There is an outer cell wall, and some move by the means of flagella. There are some forms of E. coli that will travel or swim up a stream. Sometimes they cause urinary tract infections, right? Makes sense. They swim up a urethra. Okay? They're like fish, kind of like trout. Now, some lack membranous organelles, so they don't have to have these organelles that we talk about, but some do. We don't really call them organelles at that point. The one organelle that all bacteria have that we don't really refer to as an organelle is what? Let's see if you guys can figure it out. What is the one thing that we kind of think of it as an organelle, but it technically is not because it's just peptides, proteins, and rRNA. What is the structure made of? RNA, proteins, that kind of functions as an organelle but isn't because it lacks a membrane. What is that structure I'm trying to think of? I have 6 of you. I should have 21 of you. What? Yes, 21 of you. What happened? 21. Don't fall for it? 21. What's 21? 21 vines. Sounds like a row in a vineyard. I was not born in a cave. Yeah, no, I have not seen it. I don't know. Should I watch it or no? Okay. So the fact that I'm missing some people, what's going on out there? You are not tired. You are not a car. I cannot drive you. All right, let's see what happens here. Ribosome, yay! Not mitochondria. No, believe it or not, bacteria do not have mitochondria. Okay, that's why they're only one cell. They don't have enough energy. But it is the ribosome we're looking at. So the ribosome, it does perform a function for a cell, but it is not wrapped in a membrane, so we don't call it an organelle. It kind of functions as one, but it's a little simpler. It doesn't have a membranous sac. So it may have accessory rings of DNA like plasmids. Have we talked about plasmids yet in here? A little bit when we talked about PGLO. It's more of a biotech thing. We are going to get into it more when we talk about transposable genetically mobile elements and things like that. But for right now, you need to understand that a plasmid is kind of like a baseball card for a bacteria. If I have a baseball card you want, we could trade, right? And it's an extra set of genetic material that can actually drive evolution and can drive survival. A good example of that is if I'm a bacteria and you put hand sanitizer on me and destroy me, my plasmid could be taken in by another bacteria, which could make them have my trait, okay? And if they were resistant to antibiotics and weren't harmful, but I was harmful but easily killed, now they could be both hard to kill and not be able to be destroyed by things like antibiotic or hand sanitizer. Do you get that? I'm saying that that's part of what leads to superbugs, okay? You're better off using soap and water. Okay? Well, soap, if you do it correctly, it's not only going to denature the cell membrane, because alcohol does that relatively easy, it's also going to denature the DNA, okay? Alcohol won't. It'll destroy the cell, no problem, but it doesn't kill all the plasmids and all the DNA, not kill it or destroy it. Okay? So those can easily be taken in now that they're just floating around in the environment by another bacteria that was not destroyed by hand sanitizer. And then what you end up is a bacteria that can't be killed by hand sanitizer and also now has an extra set of harmful DNA. So that's how they evolve. So bacteria can have many different structures, and I guess we'll start on the outside in. They have a capsule, which is a gel-like coating outside of the cell wall made up of a polysaccharide layer called a glycocalyx. There's a fun new word for you, right? Glycocalyx. Underneath that, you're going to have a cell wall. This is a structure that provides support and shapes the cell. So the other one underneath that all cells have to have is a cell membrane or a plasma membrane. We know all cells need a plasma membrane. If I ask you the things that all cells have, you tell me a plasma membrane, they have DNA, they have ribosomes, and they have cytoplasm, right? Those are like the 4 must-haves of any cell, and prokaryotes are no exception to that. So again, that's going to control what comes in and out. We know they have ribosomes. Nothing exists without proteins, right? So we need something to make them. And then, of course, they have cytoplasm. I told you all cells will have that. I'm trying to do all of the ones that they all will have. And then the nucleoid is basically DNA and a chromosome, so we know all cells need genetic material. And then there's like these accessory organs for bacteria. Some of them are fimbriae. Okay? These allow adhesion to surfaces. You ever wonder how a bacteria can actually cling to your cells. This is how. It's like Velcro for cells. Otherwise, they would literally just wash by your cells, and like if you had bacteria in your urinary tract, it'd be no big deal because it would just be pushed out, and you'd flush your system when you peed. We know that they like to hold on to that epithelial. It's called transitional epithelium. They can hold on to that with these pili, and they can create an infection, or they can stop sort of to set up shop. Okay, and once they stop, then they divide, and then their other buddy has pili too. And then you get a pile of pilus. That's not good. And then you get all these reproducing bacterial cells, and they're all sticking to you. Sounds bad, and it is. Then you have what are called conjugation pilus. And these allow appendages and used to transfer DNA to other cells. And that sounds really weird, right? Why would a prokaryote, and I'm going to ask you this, why would a prokaryote want to be able to transfer DNA from one cell into another? They actually share their DNA this way. You know how you put like a do and not together and you conjugate it? Conjugated pilus. Why would I want to share my DNA with my neighbor? They don't reproduce sexually, remember. Do you have a question? Oh, I'm sorry. I did not. I had like a crazy morning. Had every grandpa and grandma driving at like 5:30 this morning, I swear to God. Or 6. It took me an hour and 40 minutes to get here. Let's see. To adapt, for sure. So it's not like they sharing is caring, reproduce more successfully, to gain more for survival. So here's the kind of cool thing about bacteria. They don't reproduce sexually, but they have a lot of sexual behavior, so to speak. And when I say that, I mean anytime you're sharing genetic information, it's sexual reproduction. And I don't want you in your head to think that bacteria are sharing DNA sexually, but I do want you to think about bacteria as not being as static as we think they are just because they reproduce by binary fission, right? They still share DNA. It is the same DNA, right? But do you think each different bacterial cell experiences different mutations? Everybody say yes. Yes. Because of that, they're able to adapt and change to environments way more efficiently than we might think. Okay? Which leads to the idea of combining chromosomes, right? You guys get that? Which leads to sexual reproduction, which leads to more of a complex organism where two different cells can actually work together. It leads to the idea of colonizing and starting to create this endosymbiotic theory. Okay, I think, flagellum is the only thing I really haven't talked about. We know flagellum is like the motor. It's kind of like the tail, so to speak. It's not really a tail. It's an undulating set of actin and myosin, the two contracting proteins, okay, that basically motor, in this case, a bacteria through its surroundings. So then, on the inside, if I look at where this actual conjugation would occur, that would go all the way in, and what we're looking at here is the flagellum here, okay, and it would attach at the inside plasma membrane, the inner cell wall, and through the capsule. So it's almost like it has this barrier where it's able to be influenced by both the inside and the outside of the cell. Okay? And when it says filament, the filaments I'm referring to are actin and myosin, also made of microtubules. Okay? Very simple protein. So we know reproduction in prokaryotes is relatively simple. They are asexual, although you think about how they share through conjugation, not always as simple as just thinking about binary fission when you think about how their DNA might be changing. But prokaryotes reproduce asexually by means of binary fission. Bi meaning two, to split into two. Fission is to break apart, right? Fusion, come together, fission, separate. And mutations are generated rapidly and passed on to offspring quickly. Well, why are mutations generated rapidly? There's a few reasons. Well, let's see what you guys come up with. Why would bacterial DNA mutate rapidly? Because they're in the river. No. That was a very dumb joke. Sorry about that. The rapids. Monday, Mr. Mason. We can't handle that kind of stuff. Not allowed. Oh, there's 22 of you all of a sudden. Let's see. Okay, they reproduce rapidly, therefore more. Okay, but why is what I'm looking for. They have similar DNA. Okay. Because they divide. Oh, they have more generations, right? Think about us. We reproduce like every, maybe 80 years per person, whatever, a couple times. They reproduce hundreds of millions of times a day. That's a lot. Okay. Shorter lives. Okay, kind of like that in a way. Even if they could live a really long time, they still reproduce a lot. They reproduce quickly and often. What am I looking for? One thing I'm not seeing. Why am I not seeing it? Ah, there it is. It has no nucleus, right? So there's no protective nuclei there to protect DNA. So it's fully exposed to whatever's in the environment. So if you put hand sanitizer on your cell, and it might break the DNA in half. And if it's conjugated to another cell, it might come in through the conjugated pili. Another cell takes in a chunk of DNA. It might just fuse and combine with its chromosome and become a whole new bacterium. Yes, that can happen. Okay. And then there's one other thing through conjugation, too. You could have said that. So, definitely mutations, but also the fact that they're not protected with a nuclei, okay, and they share DNA. So prokaryotes are haploid. That means they're one N. Mutations are immediately subjected to natural selection. There's none of this waiting for mitosis or meiosis, waiting for gonads to make sperm or egg or different gametes coming together. There's none of that like accessory stuff going on. It's immediate. Whatever gets mutated gets reproduced immediately. Think about us. Males, 4 million sperm a day. And if you reproduce 4 times in your life, that's a lot. That's me twice. That means only 2 of all of those gametes through your whole life ever go into another organism. Bacteria, every single mutation gets pushed through the pipe. Think about that. Every mutation. That would be like every 4 million sperm would have a chance in the environment. Is that a lot of variation? Is that a lot of selection that the environment gets to choose from? You understand now why bacteria are so strong. Okay? I mean, we don't really think about it, but imagine if every version of you got a shot. That would be crazy, right? A lot of people, one thing. I don't think there's enough cheeseburgers in McDonald's to take care of that. So some bacteria form resistant endospores under unfavorable conditions. That means that sometimes they have these defense mechanisms where they can actually form a spore. Remember we talked about the tardigrade the other day? That's way more complex, but think about it in a more simple form where it can actually form a spore around its genetic material so that it can wait for conditions to become healthy, nice and warm and moist again. Okay? So some bacteria have the ability to kind of go dormant, almost like a latent virus in a way. Aren't bacteria fun? No, they're not fungi. No. So the method of genetic recombination in prokaryotes is conjugation. We talked about that just a little bit, but conjugation pilus forms between 2 cells. Now, the donor cell passes DNA to the recipient cell through the pilus. This part always kind of creeped me out when I learned. I didn't learn about this until college. I was like, oh, that's really weird. Bacteria almost have a sexual nature to them because they are sharing their genetic material. I don't want you to put that on a test. They're asexual, but just keep in mind that this does occur between different bacteria. And then, of course, there's transformation. We've talked about this already. How we can take a gene from one organism and put it to another, just like the jellyfish gene that we took a look at. This occurs when a bacterium picks up free pieces of DNA from other prokaryotes. So if it does take in some of this bacteria from another prokaryote, either by the environment ingesting it, trying to eat, or through the conjugated pilus, it can go through what's called a transformation, and it can become incorporated into the genome. There was a famous scientist who did experiments on this with mice. If you guys remember who that was, it was Griffith, right? You guys remember Griffith's transformation experiments with pneumonia? Yep. And then there's transduction. This occurs when bacteriophages carry proteins of bacteria DNA from one cell into another cell, and they serve as vectors. Let's talk about that real quick, because this is one that's a little foreign to us. So we talked about bacteriophages. A virus lands on a bacterial cell, okay? Kind of like the Mars rover. It lands on the cell, and it sticks its probe into the cell, and it squirts its genetic material in. And then let's say it's got an enzyme called reverse transcriptase, right? And it goes through this whole process of writing its DNA into a host cell, and then that cell blows up, lyses, okay? And then those viroids go out, and then they find other cells. In this whole process, they can actually carry and release the bacterial chromosome parts that it might have destroyed, okay? And take with it as it goes and injects another cell with now not only its DNA, but also the bacteria DNA. Sounds kind of weird, but they can actually not just transfer their own RNA or DNA. In the process of that whole transcription to that transcription process to make that RNA so it can have it in its capsid head, right? It might have some of that actual prokaryotic DNA in it too. I'll stop there. You guys, it's Monday. I know it's Monday. Otherwise, we'd be taking a test tomorrow, right? We might be taking a bacteria test.